So many thanks for sticking with this. And like I mentioned at the beginning, I'll talk a little bit in the second half about technical challenges now around defining defining a healthy microbiome and measuring the microbiome in general. So to start with a quick, very basic rundown of more or less the modules that lead from a biological microbial community in the wild down through having features of the microbiome that we can put into a definition of healthy. I would list these as sample acquisition, how do you get bugs in the first place, sample handling and storage, what do you do with them after you have them before generating data, a series of more cellular assays that can take place at this early stage, which include live cell-based methods, culture or notobiotics, whole cell imaging, for example, or if you're going on to molecular data for a definition of a healthy microbiome, we need to get molecules out of a microbial community somehow, be they nucleotides, DNA, or RNA, or other molecules such as proteins or metabolites. All of these need to be ripped out of the containing microbes one way or another, assayed through sequencing or otherwise, the otherwise is usually, I'd say, a mass spec if you're not doing sequencing at this step, in addition to, say, NMR metabolomics, a couple of other options here. And then downstream of generating molecular data, there's some series of computational methods to manipulate those data into interpretable features and some statistics to manage those features and compare them or assess them across a population. So you can think of each of these as a module where things can go right, things can go wrong, or you can introduce technical variation, be it good or bad. And it's surprising in microbiome studies how much technical variation can be introduced at essentially every step in this pipeline, more than most other molecular omics assays, in large part because different bugs are different. The heterogeneity, even within a single community of individual microbes, manifests in surprising ways at different points along here. So to take one example, using two very slightly different swabs, for example, a cotton swab versus a nylon swab, to measure exactly the same microbial community can sometimes generate very different assessments of which bugs are there. Because within that one community, you have microbes with all different cell wall compositions, some of which might stick just a little bit better to nylon, and some of which might stick just a little bit better to cotton. So this one apparently tiny technical choice, because of the heterogeneity inherent in each microbial community, can generate unwanted technical differences by the time you get to the end of your measurement. And then, of course, each technology that you use for these different assays can have its own sort of biases or tendencies in generating measurements. And an easy one that I think is most pervasive in the field is the sort of different technical effects of Amplicon sequencing, 16S sequencing, versus shotgun sequencing. So an easy example, they're both affected by differences in how one extracts DNA from a microbial community. But Amplicon sequencing can then additionally be affected by things like primer biases or contamination that's amplified during the extra amplification step that won't necessarily be shared with shotgun metagenomics downstream. So both the underlying biology and the individual technical steps can manifest in noise, essentially, in sometimes surprising ways. And of course, we don't want this. If we want to define a healthy microbiome, it needs to be consistent across populations globally, as Claire was saying earlier, and across measurement technologies and different studies leading up to that definition. These different assays can yield, again, kind of a surprising range of different, not yet really standardized microbiome feature types that could be used individually or jointly in a definition of health. And these range from different ways of defining microbial taxa, the composition of a community, through different ways of defining the functional composition or readout of community molecular activity, and everything in between. I didn't really go into the details in the last talk, but there are multiple different ways, of course, of defining a microbial strain, even if those are in some ways the fundamental unit of what makes up a microbial community. And of course, each of these different feature types have different strengths or weaknesses, and different measurement error that leads into their definition and then can lead out of these definitions into downstream statistics and comparison between studies. So to give one quick concrete example of this, I'll draw a few examples in this talk, mostly from a study that we worked on called the Microbiome Quality Control Project, or MBQC, where there were many people involved. Scott and others have continued this work more recently. But now, several years ago, this was one of the first large-scale studies to look at reproducibility and sources of technical and biological variability in human microbiome population studies. Starting with a subset of the potential noise points that I mentioned a few slides ago, specifically the subset of just within gut 16S amplicon sequencing of sample collection, extraction, sequencing, and bioinformatics. And this was inspired in part by an example that I'll draw from the Human Microbiome Project again, where in early comparisons with MetaHit, one of the other large gut microbiome population studies, even forgetting all the complexities of these populations, it's easy to see at a very high taxonomic level that there's an enrichment for blue, which happens to be phylum Bacteroidetes in the Human Microbiome Project, and enrichment for orange, which happens to be Firmicutes in the MetaHit. And maybe a couple years after the first papers on these big cohort projects came out, there was a really nice follow-up study showing that this isn't really a biological effect. This is a technical effect having to deal with mostly different extraction kits that were used between the two projects. So it's easy to blame this entirely on biology until you dig into these individual technical steps that can also feed into differences in feature measurements. So the design of the MBQC itself was a volunteer effort, so we had to be very focused in picking apart just a few of these potential sources of variability. We looked at a standardized set of 22 different specimens, original physical specimens, some of which were either fresh or freeze-dried stool, several of which were control specimens, either replicates of the same complex composition from a chemostat, replicates of the same ground-up, entirely defined positive control community, or negative control Blake samples. These were replicated and triplicated. Some of them were centrally extracted. Some of them were sent bulk. And then again, some of these were controls to make up a single 96-tube sample set that was blinded and sent to a total of 15 different participating labs for generating data. These data were then again blinded and randomized and sent to a total of 9 participating labs for downstream bioinformatics. And again, I put this up here and sort of stall on this slide because it was all volunteer effort, and I really want, every time I present this, I want to thank everyone who participated in it over the course of multiple years. One of the things that's remarkable, this took place in about 2014, 2015, which was maybe 3 years after the Human Microbiome Project 1 was published. And running on a shoestring, almost $0, we ended up generating about 2,200 samples worth of sequencing and a total of 16,500 analyzed samples, which is around 6 times the number of sequences coming out of Amplicon sequencing in the HMP. So sequencing really is cheaper than it used to be. It gets a little better every year. So coming out of these 15 labs times 9 different analyses times 96 replicates times originally 22 samples, we get an overview of different sources of variation arising at a high level by which lab handled the samples and generated data, and which labs analyzed those data. And so if we start even at just a very, very high level of those 16,500 samples that made it all the way through analysis, you can find examples in this overview ordination of large variability introduced by each one of the possible sources, be it biological or technical. These little clusters that you see around some of the outgroups are biological variability. These are samples corresponding to the original, one of the 2 original positive control mock communities that was constructed like an oral community. This cluster corresponds to the second that was constructed like a gut community. These are 2 real physical biospecimens that happen to come from very different individuals. The bulk of the physical biospecimens are over here. But if you think about this amount of variation, we can see similar amounts of variation, for example, in this cluster of blue points, which correspond to one particular bioinformatics pipeline for handling the data downstream, or in this little cluster of J's right here, which corresponds to one particular data generation lab that happened to have a particularly different protocol. So each of these potential sources of variability, be they analysis or data generation or the originating biology, were of comparable effect size, kind of like we were saying earlier for, say, antibiotics versus race ethnicity versus dietary components. They're all large, but they're kind of comparably large. In this case, zooming, I won't go into the MBQC in a lot of detail today, but the two most useful areas to zoom in on during the analysis were on the positive and negative controls. They allowed us to see with a lot of clarity where the sources of variability tended to be coming in. And I want to thank Emma Allen-Vercoe's lab for generating these. There were combinations of gut-like and oral community-like microbes that Emma, of all people, was amazingly able to culture and get these into realistic microbial community mixes. This then gave us a gold standard for the two positive controls where we knew what the right answer was. So we could measure not just variability, but variability relative to a standard, essentially error. And this led to a spread across, in this case, the 15 different data generation labs that summarizes, again, all the other potential variables. The y-axis here is essentially distance from truth. So there was a group of about 6 labs that were comparably good, essentially. They were close to the truth, both for the oral and gut communities, and whether those samples were extracted locally or in the centralized DNA extraction that we did for some of the samples. In the other half of the data generation protocols, anything was possible. There were cases where local extraction tended to be very different from the centralized extraction process. There were cases where the oral versus gut, the oral versus gut communities tended to be affected differently by that protocol. And there were cases where something was just generally very different. And we can see a little what that was on the next slide with the negative control. The same thing tended to happen for different computational protocols to a much lesser degree. You'll see that the y-axis for the similar summary of computational variability is shorter, is half the size of the data generation distance from truth. But otherwise, the same different combination of effects were in play. So surprisingly, we could find a few cases of interaction effects between, say, how you extracted a microbial community and which computational pipeline you used to identify which bugs were there. And this always comes back to the fact that every individual microbiome contains a huge diversity of bugs with different cell wall structures, with different genomic architectures, and it's hard to make a one-size-fits-all pipeline. From looking at the negative controls, we were able to see more clearly that one of the more common sources of variability was different types of contamination. So we included a couple replicates that were just buffer blanks in each of these tube sets. And what you're looking at here is a readout of which taxa were detected in each combination of handling lab, data generation lab for these blank samples, and that's per panel. And then within handling lab, each computational bioinformatics lab, and that's per column. So you can again see that the larger source of differences in negative controls was handling lab. One panel tends to be more different from another panel. But within panel, even for negative controls, there are differences in exactly which bugs were detected based on how you process those data. And when we zoom in a little bit more and summarize these, we can kind of guess where some of these different negative control contaminants might have come from. So each column here summarizes a whole panel. Each column is one whole data generation lab. Each row is a zoom in on the most abundant couple of taxa up here, some of which are, if we just look down the top, traceable to contaminants in the originating lab, so bugs that were being grown in the lab that handled these microbiome samples, bugs that are probably coming from other gut microbiome samples, so contaminants from the rest of the sample set, so many of these are typical gut microbes. Pseudomonas, which is a typical reagent contaminant, showed up in a couple of the labs. And then in one, we had an example where there's probably some skin contamination into the negative controls. And so this is just one source of one type of variability, I should say, coming from contamination into negative controls. It turns out these gut microbial contaminants were one of the major sources of variability in the positive controls as well, but just the diversity of different types of microbes in the negative controls and the different places they probably came from is indicative of where a lot of the other more technical sequence variability and barcode variability and bioinformatic variability came from as well. So again, I don't want to get into the details of the MBQC in particular today, except to show it as one example of a study that touched essentially the range of different protocol variation for microbiome data generation and handling, some of the sources of variability. And you can look at this in either a glass half full or a glass half empty way. The glass half empty way is that, oh my goodness, they're amazing, so many sources of variability. They're of comparable size to the biology, which can make them challenging to handle. It is difficult to resolve microbial community measurements from different protocols. But the glass half full way is that most of the time, even in this highly constrained experiment, biological effects were greatest. So on average, the differences between biospecimens were the largest measurable signal in the data, essentially, followed by differences in some of the data generation variables, especially because this was Amplicon sequencing and nucleotide extraction, the rest of the sequencing protocols, differences in computing, and then a baseline level of noise, essentially, that was present even in technical replicates. So keep this in the back of your mind again as far as technical sources of variability in defining microbiome health. Where this tends to manifest when considering population studies is that it can be very challenging to compare microbiome studies even from similar populations, let alone, as we saw earlier, from different global populations. So biological sources of variability between study populations or technical sources of variability might not mask the effects of a microbiome perturbation of interest within the study if that perturbation is still, as we saw earlier, the largest source of biological variability, but it can make it very difficult to combine those studies. If you essentially just overlay them blindly, you get a gamush rather than consistently meta-analyzing out the technical effects while keeping in the biological effects. And this is a cartoon, but you can see this kind of challenge in essentially any meta-analysis that starts to combine multiple different studies of the microbiome, even in a fairly constrained biological space. So this is an example drawn from 10 different studies of the inflammatory bowel disease microbiome, totaling around 5,000 or so samples, in which if you look at these colors here, don't worry too much about the contents of the population, and certainly not about the microbial community configurations. But if you just look at the overall spread, I can tell you that this is the group of most active disease subjects. So again, biology is the largest effect in the data. That's good. But across the rest of this sort of control population, some of what the spread in the data is biology, is underlying differences in mostly Bacteroides to Firmicutes ratio. But then that's confounded with differences between study and exactly how these samples are generated. Some of them are biopsies, some of them are stool samples, some of them were extracted using the same protocol, some of them were not. So this is representative of what happens when comparing real populations that might have a mixture of shared biology and shared technical effects in the data. That's again doing this sort of glass half full, glass half empty, glass half full. That's the challenging part. That's the glass half empty part. On the glass half full side, there are other types of microbiome meta-analysis that don't have quite as many technical hurdles. So it can be very difficult, as we just saw, to compare microbiome studies that rely on quantifying the abundance of specific features. That tends to be what is most affected by the technical effects that I described in the previous few slides. There are other types of microbiome features, such as defining the phylogenetic placement or presence of a specific strain, that are much less susceptible to technical effects. So to again draw an example from real data, we're just finishing up a meta-analysis of a different set of about 10,000 shotgun metagenomes drawn from around the globe, focusing on strain composition rather than feature abundance like we saw in the previous slide. And here there's some fairly serious computing that goes into just processing 10,000 different metagenomes. But what falls out at the end is a set of about 4,000 to 5,000 species-level, genetically defined, essentially strain-level species-level bins that contain strains, I should say, that we can place phylogenetically, that we can trace back to individual samples, and which, to go back to Claire's original point, one of the major drivers is essentially the development of the environment in which these samples originally came from. So are they coming from essentially developed or westernized populations, or are they coming from more international populations? So I got lucky with the sign again to wrap up the second part more or less on time here. One thing I think will be good to talk about for the rest of the day will be the different feature types, the different measurement types of the gut microbiome that can be used in definitions of health. But whatever either the definition is or the feature types going into it, they should be consistent across sometimes highly diverse global populations and across the measurement technologies used to study those populations. And this is currently difficult but not impossible due to this technical heterogeneity, the differences in protocols available to study the microbiome, and the underlying cellular diversity. Different bugs are different, unlike a lot of, say, human DNA that can make it easier and more tractable to measure carefully. So within this diversity, there are certain methods like strain typing that are generally less variable across populations. As I mentioned in the first half, we don't necessarily understand these biologically as well yet, but technically they might start to help tackle some of the variability in measurement technologies across populations. I also have some considerations for the short and long-term goals that we can bring up later during the discussion section, since I'm about out of time. Two things that I want to mention just at the top, because they haven't really come up yet, is that I think there is a short-term opportunity in really establishing the language around what we mean for microbiome health, let alone the scientific definition, but just what this might mean in terms of this underlying biological variability. And this came up a bit in Barbara's talk, but I think this language definition extends especially to how we, as a community and during public outreach, discuss microbially associated food products, be they live cell probiotics or microbially related prebiotics and food composition. Even outside of defining what is healthy, I don't think we have a consistent language in the community to discuss it yet, and certainly to explain it to the public. The rest of these are more detailed, and we'll get into them later, because anyone whose face you saw on the slide was the one who did the work. I want to especially call out Nicholas Agata, who has his own lab now and has done a huge amount of this work, Wendy Garrett at the School of Public Health, and Romnick at the Broad, with whom we've done several of these studies. So thanks again.